Hi, everyone. Today we are going to do a quick introduction to a variety of types of graphs that we are going to use as part of what is called exploratory data analysis. And when we first get our data from our experiment or from our study or whatever we've been collecting data from, like we talked about before, our brains do a very bad job of looking at a long list of numbers and seeing any pattern in it. So we want to create a lovely visual summary of our data. So we're going to make graphs or charts of it. Depending on the kind of data, there's going to be a correct or proper way to show those data on a graph. And that's going to depend on what type of variables we have, whether they are quantitative or categorical, and what pattern or relationship we're interested in looking at between them. We're also going to talk about not just visual summaries today, but just calculated descriptive summaries. You may already be familiar with the idea of the mean or the average. So we will talk about those types of descriptive summaries as well and what they tell us about our data set. All right, so the major types of graphs that we use in biological sciences include the histogram that we've already talked about, a bar chart, a pie chart, several kinds of line graphs, a kind of graph called a box and whisker plot, and then a scatter plot. Each of these types of graphs, like I said, is specialized for a particular kind of data. So remember that we have categorical types of data that are discrete or separate kinds of things. We want to put our individuals into a category. We may have more than one category that we want to put them into. We want to say that this is a student who's a biology major who's planning to transfer to SDSU versus a chemistry major who's planning to transfer to UCSD, right? So major and transfer institution are those two different categorical variables. So we're going to count up the number of individuals there are in each of those categories. We might make a bar chart or a pie chart of those kinds of data. A line graph, on the other hand, is going to have either a categorical variable on the x-axis or a special type of categorical data, which is ordinal. So we might have a time series, different years in a row. That's the most common kind of line graph. Typically, we're going to put a line connecting together those time points. Whereas if we're just putting a mean on a graph and saying, oh, well, of this kind of thing, my average was this, and this other mean I took over here was this average, we aren't going to connect those together with a line, and I'll show you what that means in a little while. For these line graphs, we're going to have on our y-axis some kind of quantitative variable, something we measured numerically. Now that thing, we might start out by making a histogram of it, and then we might also put it onto a line graph to compare two different groups or types of individuals. We can also summarize our data set using a box and whisker plot, and I'll show you how we do that. It's kind of like a histogram on its side, and we may use that in certain circumstances to look at our distribution of our data, not just the mean. And then a scatter plot, we're going to come back to in the next lab. I'm going to spend the whole lab working on those. And they're going to have a quantitative variable on both their x-axis and their y-axis, right? So we have these different types of graphs, and which type we use is going to be determined by what kind of data we have, whether it's categorical or quantitative, right? And so if we have only one quantitative variable, we're going to make a histogram, maybe a box plot of it. If we have, in addition to that one quantitative variable, we have a categorical variable, that separates our individuals into different kinds. We might make a line graph. And if we have an ordinal variable, we might plot it as a time series. If we have two quantitative variables, we want to know if they're associated with one another. That's going to be a scatter plot. And then if we have one or two or more categorical variables, we'll use a bar chart. So we also want to keep in mind that the variable along our x-axis is going to be the one that we consider explanatory or independent. That means that we think something about that variable is going to cause or be responsible for the thing that we're measuring that we're going to put on the y-axis. So that y variable is going to be called the dependent variable or the response variable. Right? So explanatory versus response is the way we may talk about those axes in statistics. All right, we already looked at histograms. We're going to have a continuous x-axis that should be labeled, and then how many observations we have in each of those bins. That is going to be different than a bar chart. So our bar chart is, instead of having continuous variable across the x-axis, we're going to have steps or categories. So in this graph, it's showing for all of you smart people who are working on improving your education, that should end up resulting in a higher mean annual earning. So we have education, highest level education level attained along the x-axis, and then we have on the y-axis median annual earnings in dollars. And we have a little gap in between these bars. Remember, on our histogram, we don't have a gap between the bars. Right? No gap on the histogram. We're going to have a little gap between the bars on our bar chart, just like we do on our probability distributions that we made at the very beginning class, because there isn't anything in between them. All right, now we could make this graph differently. We could make it based on, say, years of school rather than looking at specific step levels like we did here. And in that case, it would not be a bar chart. It would have a continuous x-axis. All right, so here we only have one variable. We don't have males versus females. We don't have race or ethnicity differences. We just have everybody lumped together. All right, but we may also have information that we want to show that has not just one categorical variable, but a second categorical variable that goes along with it. So this is a fun data set that shows our income of individuals, whether it's above average, average, or below average, and whether those people report themselves as being not happy, pretty happy, or very happy. So our overall pattern that we can see, we want to compare happiness across these income levels. And so we sort of clustered together these happiness levels. And we actually see about the same pattern for each of these groups, that the most individuals, whether the income is higher, low, or average, most individuals are pretty happy. However, we see a little different pattern when we have when we look at the very happy relative to the not happy, right? Above average incomes, we have more people very happy compared to not happy. And that is also the same for average incomes. When we get to below average incomes, that pattern is flip-flop. So we have more people who are not happy compared to very happy. Right, so this overall pattern shows us that money doesn't buy happiness in general. All right, so let's look at the data that we use for making this graph and think about how this graph looks and how we have the number of respondents here, people who responded to our survey. So there were fewer total people who responded to our survey who were in this above average category. And what consequence does that have for our graph? All right, so we're going to summarize kind of data that we're going to show when we have these two categorical variables. It's going to be called a contingency table, right? We use that word contingency to mean what are we going to do if something happens? Well, my fallback plan or my plan B is often referred to as a contingency plan. A more general definition of the word is that it's when something depends on something else. It is contingent on it. Right. So our contingency table or our two-way table is going to let us know, based on various calculations we can do, if one of these kinds of things depends on the other. Right. Does happiness depend on income? Right. So we're going to send our survey out. We're going to ask people what their income is. We're going to put it into these categories. We're going to ask them how happy they are. We tally up how many people responded in each of these two-way categories. Right. So notice that we have the fewest total people in our above average categories. So we're going to do some summary statistics on our contingency table. We're going to add up the row totals. We're going to add up the column totals. Our total tells us the sum of all the total individuals that we asked in our survey. And it's going to be the sum of either our row totals or our column totals. Those should come out to add up to the same thing. All right, so suppose we want to make sure we're comparing just proportions out of our group instead of counting up individuals, right? How does 21 compare to 143, right? We want to look at those numbers scaled for the sample size in each of those income categories. So we can remake this graph. It'll show that pattern more clearly if we calculate some proportions rather than the counts. All right, so we can take 21, divide by 360, 213, divide by 360, 126, divide by 360. That's going to give us our proportion of people who had above average income, that were not happy, pretty happy, and very happy. And we're going to do the same thing for our other rows. 96 divided by 850, 506 divided by 850, 248 divided by 850, and the same thing for our last row. All right, so if we look at the graph, we're going to see that it looks a little bit different. All right, we have proportion of respondents on the bottom. The other original graph is shown again at the top, has number of respondents. So we should see the same pattern, right? We still see that there is a higher proportion that are very happy compared to not happy in the above average income. But it sort of zooms in on that pattern and it lets us see it a little more easily. Right? So this graph on the top isn't wrong. If we look at the proportions instead of the counts or the relative frequencies, we maybe can see that pattern a little more clearly though. All right, so we'll do this kind of calculations. We come back later in the semester and we're going to do a kind of test called chi-square test, which is going to let us see if, in fact, the distributions that we see between these different income levels are actually significantly different from one another in their pattern. Right now, we're just thinking about what we're showing on the graph, what the data table is going to look like in Excel, and how to summarize what these graphs are telling us, right? So if we look at this lower one again, we see that the proportion of sort of moderately happy people is about the same in each income group, but that we have kind of as income goes down, we have fewer very happy people and more not happy people. All right, so those are bar charts. All right, the same type of data we can actually use to make a very similar graph that shows proportions called a pie chart. I love this picture, the world's most accurate pie chart. Pie I have eaten versus pie I haven't eaten or that I will eat. And our pie chart is going to just show 100% in a circle and then each proportion as a wedge taken out of it. Business folks use a lot of pie charts. Biology people don't use them as often, but there's one area where we do use pie charts a lot, which is in genetics, because when we look at different genotypes, different alleles in populations, we sample the whole population, we're showing 100% of the population, and we can show the distribution of genotypes in across different populations really quickly and easily. So this is a lovely figure showing genetic information of Y haplogroups. So these are from Y chromosomes on genetically male individuals in Europe. And these different haplogroups or genotypes are just different recognizable DNA sequences. So when we look at the pattern we see across Europe, we can pretty easily see, based on these nice diagrams, that the frequency, the proportion we find in the population of this haplogroup represented by the red color, R1b, has very high proportion when we look at the far western edge of Europe, and then it decreases in frequency as we go to the east and to the north. Right? And other haplotypes are more common in different geographic areas. And these pie charts are a really nice way to show those differences in proportions. All right, our next type of graph, we're going to look at a line graph, and that usually is going to be some graph where little dots are connected together with a line, and it's going to show a pattern over time, usually. So we're going to have year, for example, for this graph. This is a special graph. It's called the Keeling curve. It's not really a curve. It's a line graph. It's showing carbon dioxide concentration in parts per million on the y-axis and time in year on the x-axis. And it's showing carbon dioxide concentration is fairly steadily increasing. There's these little wiggles that occur in inside every year. So carbon dioxide concentration gets higher in the northern hemisphere winter, when the vast forests of the Canadian region and Russia are asleep in the winter, and they're not sucking up huge amounts of carbon dioxide, so all the animals breathing out increases global carbon dioxide concentrations in the northern hemisphere, and then the plants wake up in the spring and summer and they suck down all of that carbon dioxide and turn it into more plant. And so we have these little wiggles that represent winter versus summer, and then this is steadily increasing as it goes from about 1960 to about 2020 here. So that's a very famous time plot. It's over a relatively short time period when we think about the history of the Earth. So when we look at these kind of data, we might want to put them into a broader context. Here's another graph. This one's from NASA, showing carbon dioxide concentration, again in parts per million, but going back 400,000 years. So we see that our carbon dioxide concentration that was in about 1950 is where that falls relative to historic or paleoclimate, and that if we look at the large differences in carbon dioxide concentration, it's been much lower, but through this period of time, it hasn't ever been higher than it is now. So when we're looking at a time plot, whether we're super zoomed in or whether we're super zoomed out, we're going to be able to see a pattern in our data that we might need to see a larger time frame of it in order to put it into context. All right, so when we're looking across, so we have a single quantitative variable that we're plotting, and our time plots are going to give us an answer to the question, is something changing over time? Is this variable increasing? Is it decreasing? Now remember, we looked at histograms. One of the things we used them for was to evaluate whether there were any outliers in our data set, unusual observations. Okay, so if we look at another example of a histogram of the data, so this is our Mississippi River discharge in cubic kilometers, and the count or the frequency that amount of river discharge, so how much water came out the mouth of the river. And on the right-hand side, we have a time plot of the same data. So in the graph on the right-hand side, we have associated each data point, each quantitative value with its year, and we have put them in order on our graph. All right, so some years we have lower amounts of river discharge, some years we have higher amounts of river discharge. And the reason we might look at a time plot is to see if there's a trend, right? Is it going up over time? Is it staying the same, or is it going down over time? Now on this graph, if we sort of squint at it, it might look like it's, going up over time. We have to do an analysis in order to make that evaluation. And we see some kind of high points up here. Are any of those points unusually high or unusually low? Well, we can know that by making our histogram, right? And we look for outliers. And if we had a value that was, way over here, like 1,200 cubic kilometers, right? Or if we had one that was way over here, 50 cubic kilometers, and we saw it on our graph, it would be a value that was like way above these observations or way below them. So from looking at the histogram and the time series, right, we can say, well, there doesn't seem to be any really unusual years here. And maybe it looks like there's a slight trend going up, but then we have this, value over here that's low again. So it isn't really consistent. It's maybe we have a slightly different, maybe something happened between 1965 and more frequently, but that's about all we can say right now. Whereas if we are looking at that time series of our atmospheric carbon dioxide concentration, there is a very clear pattern that it's just continuously going up. All right, so for a time plot, we're going to have years along our x-axis and some quantitative variable on our y-axis. All right, so before we talk about the next couple types of graphs, we need to talk about calculating our descriptive summaries, because sometimes we want to make a graph that doesn't have all the data on it, that it actually just has the mean on it. And we always want to report not just the mean, but a value that lets us know how much variation there is around that mean. So we're going to talk about calculating a couple different kinds of these descriptive summaries, the mean or arithmetic mean or average. And we're also going to find a quantity that's called the median. Median is defined as being in the middle of the data set. If our data is normally distributed, the mean and the median will be the same number. And so if our data set is perfectly symmetrical, then mean and median will tell us the same thing. The standard deviation, on the other hand, is going to tell us how wide our histogram is. Is it a really skinny histogram where all the data is clustered very closely to the mean, or is it a very wide histogram where we have lots of variation between individuals? Right. So the standard deviation is going to be a measure of how variable our individuals are from one another. Standard deviation is going to go along with mean. We have a similar measure called inter-quartile range that's going to go along with median, and that's also going to tell us how wide our data set is. So when we think about these two different kinds of measures, we're going to call the mean and median measures of center or location. They're going to tell us where exactly the middle, but the sort of center of mass is of our data set, and this is a quantitative variable. Where is it located on our x-axis? Is it over here? Is it over here? And it's going to give us a place where we can sort of say, my data is right around here on this x-axis. These other measures are measures of spread or dispersion. How different are my individuals from one another? And inter-quartile range and standard deviation are two of these, but there's also two more. we can just say, well, how big a difference is there between my largest and smallest values? And that's called the range. Standard deviation, we're going to use a character to denote it. We're going to use little s for standard deviation. And we also have a measure that's very similar, but it's going to be calculated as s squared. So it is literally the square of the standard deviation. It has a name. It's called the variance. The mathematical properties of standard deviation and variance are slightly different. Some kinds of calculations we want to make, we have to use the variance because of the rules about how it works mathematically. Sometimes we can use the standard deviation, so we need to know them both. All right. So we're going to start by looking at mean and standard deviation. And if our data set is normally distributed, the mean is going to be right in the middle. It should be the dead center of this highest bar. And then the standard deviation is going to stretch out to the side. So when we say that the mean is a measure of location, this figure is sort of saying, well, this is where my data set sits on my x-axis, whereas standard deviation and inter-quartile range are going to say how stretched out across my x-axis are all of my data. All right, you have probably calculated mean before. We're going to use a formula to show how we do that. So to find the mean, you know probably already, you add up all the observations in your data set, and then you divide by how many observations you have. Now, we're going to put that into formal mathematical notation, and we're going to call the mean x bar, and it's got a little superscript bar across the top of it, so little x bar is going to be our symbol to denote mean. And that dividing by the sample size, we're going to call the sample size little n. All right, so for n, starting with 1, 2, 3, whatever, how many observations we have, all the way through little n. We're going to take each of those observations, the x sub I, the x sub little I, right? And we're going to add them up. So our summation sign is saying from 1. Where should my summation start? We're always going to start it from 1 in statistics. And it's going to go all the way to n. All right, so why do we bother writing this? Because we're going to get some much more complicated formulas as we go through the semester, and understanding the notation when we're using a very simple formula that you understand already is going to help you get used to this notation. All right, so we're going to take all of our observations across all of our different individuals. We're going to add them all up, and we're going to divide by the sample size, and that is our mean x bar. So let's work through a quick example. Now, with all examples in this class, you should definitely type these into your calculator and make sure that you get the same answer that we are getting as we work through the example together. There's a few things that you can do in your calculator that sometimes are going to give you the wrong answers. So you want to make sure that you're using your calculator correctly and that you're not making those mistakes. You can only figure that out if you do the practice problems. All right, so we've got a small data set here with 7 individuals, right? These are different individuals. We measured their size. I think these are sardines, small fishes, and we measured them in centimeters. Notice that our data has 1 digit after the decimal point. All right, so first we want to start by finding little n, our sample size. We have counted up the individuals. And so we know that our sample size is 7. So we're ready to plug that into our equation. The next thing that we're going to do is identify each of these little x sub i's, right? And we're going to cycle through all of the little i's, right? What are our little i's? Little I is standing in for all of the different values of 1 through n. All right, so we're going to start with our little x sub 1, right? Because we're summing up. Our little x sub 1 is going to be the value that goes with the first individual, and that's going to be 6.3. And then we're going to take the little x sub 2, right? We're going to cycle through all of our little our values for little I. All right, so then we're going to add 13.8, so on and so forth. We're going to show that in the next slide. All right, so here I've taken my 1 divided by 7, and then I've added up all my observations all the way across. Right, you should get a sum of 60.1. Don't forget to divide it by 7. And this notation, it should be x bar, little x. All right, what do we get? We get 8.586, right? Remember when we talked about significant digits? We're going to report our result, our final answer with a nice box around it. We're going to round off so that we have the same number of digits that our data had. And we're going to round off so we have the same number of digits that our data had. And our answer for what our mean is should always be reported with its units. What is this thing that I measured, right? We have to give you the units, centimeters in this case, right? So my final answer, 8.6 centimeters. Standard deviation is a little more complicated. We have to find the mean first. Right? We can't find the standard deviation unless we've already found the mean. And we're going to start talking about finding the standard deviation using this kind of bulky looking equation. So remember, standard deviation is going to be little s. And we're going to go all the way to the middle of this equation. So remember from your algebra, you're going to go all the way to the middle of your equation first. Again, we have these x sub i's, right? So we're going to take each of our x's, each of our observations 1 through n, and in turn, we're going to subtract the mean from it. Right? We're going to subtract the mean from it. Now, the definition of the mean is going to be that we're going to end up with half our observations above it and half below it. So if we subtract, we're actually going to get some negative numbers, we're going to get some positive numbers. And if we just added those things up, they would equal 0. So we have to do something in order to not make that happen. And the thing we do is that we take these values, these deviations, all right? So each observation minus the mean is called a deviation. We're going to take these deviations and we're going to square them. What does that do? Well, it turns the negative ones positive, right? So when we add them all up together, we get a positive number. All right, so we're going to take each of these of our observations, we're going to subtract the mean, we're going to square it, we're going to add them up, right? Just like we did for the mean itself, we have the summation from 1 to little n. And then instead of multiplying times 1 divided by n, we're going to multiply times 1 divided by n minus 1. N minus 1 is not the sample size, it's the degrees of freedom. Right? Degrees of freedom tell us how many independent observations we have. Once we've calculated the mean, we have little n minus 1 degrees of freedom because we have all of our little n plus the mean. If we have, so we actually have n plus 1 pieces of information, we can always calculate the last observation if we also know the mean, right? So we just took all of our observations and we used them to calculate the mean. Once we've done that, we actually only have n minus 1 independent pieces of information, right? If you don't really understand that, that's fine. Just know that in your standard deviation calculation, you should have n minus 1 in the denominator instead of just little n, right? Some calculators are not going to use n minus 1. You want to make sure that if you're using a standard deviation function in your calculator, that it gives you the right value. Right, so double check that. Now, we squared x sub I minus x bar. If we measured our fish in centimeters, that means we're going to have centimeters squared as our unit here, right? We don't want to, that's not how we want to present our result. So we're going to take the square root of the whole thing in order to get back from centimeters squared to centimeters. All right, so this looks like a long and complicated formula. by the end of the semester. It will not look like a long and complicated formula. And we're going to go through an example again to show how you do it step by step. All right, so same data set. We've already calculated our mean, 8.6. And we're going to start by plugging each observation in and finding these deviations. All right, so we're going to take the square root of 1 over 7 minus 1. And then we're going to take each observation, 6.3, subtract the mean, 8.6. We're going to square that. And we're going to add it to 13.8. We're going to subtract the mean. We're going to square it. We're going to add that term to the next observation, 5.7. Subtract the mean, square it. All right, blah, 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 all the way through the data set. And this square root is encompassing all of these terms. So we want to do all the squarings and all of the addings. And then we're going to divide by 6 and take the square root of the whole thing. So we'll go through the rest of the steps in these next couple slides. All right, so this is going to simplify to 1 divided by 6, right, times 134.49, and you're going to take the square root of that whole thing. And so make sure for all your terms, all these subtraction and squaring terms, that you end up getting 134.49. All right, so if we do this division, we're going to get 22.415. We still need to take the square root of it. Don't forget to take the square root of it. Right, we're going to end up with 4.73 centimeters. All right, so for our standard deviation, what does this tell us? All right, this tells us that we have a mean of 8.6, and our standard deviation is 4.7 or 4.73. 4.7 is pretty big, right? It's about half of the value of the mean. That tells us our data set is pretty variable, right? If our standard deviation was 0.2, right, that would tell us all of our measurements are very close to our mean. And if we look at our data set, we have values going all the way from 4.7 up to 16.8. So it's pretty variable, right? So we're usually going to want to look at our standard deviation relative to the size of the mean. It's going to give us an idea of whether our data set is highly variable, the standard deviation is large, or if it's not very variable at all. Most of our individuals are very close together. Right? So if our data set was like all of our observations were like 7.9, 8.5, 8.7, you know, there was maybe a 6.9 or maybe a 9.0, right? Then we would have a much smaller standard deviation. And there's a bunch of reasons why we want to know that, but the simplest one is just reporting the mean provides us with a good deal of information, but reporting the mean and the standard deviation not only tells us where the data set is along the x-axis, but some idea of how variable it is. So usually when we are reporting the results of our experiment and we're summarizing the data, we're going to say this is the mean and this is the standard deviation. So these pieces of information go together and they should always be reported or shown on our graph in some way. All right, so median and interquartile range are similar measures, but they're found in a very different way. And what we're going to do with our data set is first, we're going to take our data set, same one we had before, and we're just going to sort it into numerical order in order to find the median. The median is just the middle observation. So when we rewrite our data set, starting with the smallest and going to the largest, this data set happens to have 7 individuals in it. So this middle one, the 4th observation, that's our median, 6.3 centimeters, right? So there isn't really a formula for finding the median and the quartiles. We're gonna just sort of order our data and then we're gonna find it by hand. So it's the observation that's in the middle. Half of the data set is smaller than this value. The other half of the data set is larger than this value. That's all the median is. The quartiles of the data, right? Our median is basically the 50% mark. If we take 2 quarters, we get a half, right? So the median is also the second quartile, the second quarter or half. Similarly, we can take the lower half of the data set and find the observation that's in the middle of it. That defines the first quartile. We're a quarter of the way through our data set. And then same thing, top half of the data set, find the middle observation. That gives us the third quartile. We're 3/4 of the way through the data set. All right, so we found our median and our first and our third quartiles. We want to have some measure of how wide the data set is. If we're using median, then we're going to use inter-quartile range. That is the distance in between the first and the third quartiles. All right, so once we've found our first and our third quartiles, we're going to take the third quartile, we're going to subtract the first quartile from it, and that's going to give us the IQR, inter-quartile range. All right, and in this case, it comes out to be 13.8 minus 5.5, or 8.3. So we would report the median of 6. What did we get? 3, and the interquartile range of 8.3 as equivalent to the mean and standard deviation of our data. Right, so when we calculate the mean of a data set, we may be able to make a calculation that gives us an answer that's quantitative, even if our data are ordinal. So if we want to find the mean, say, of how many children families have in the United States, we can get this weird, because it's ordinal data, we can get this weird answer where, for example, we can get a family with 2.5 children as our mean. Well, does that mean that any family actually has 2.5 children? No, but it means that, you know, about half of families have more children than that and about half have less, right? So this cartoon is a little hard to see. Here's their 2 whole children, and this is a half of their other child. It's only the bottom half. This child has been chopped off. All right, so that's a funny side note about how averages can work sometimes. All right. So suppose we were doing some kind of experiment or study where we went out and we measured 2 kinds of things, and we wanted to say how big they are compared to one another. This is a very common type of data that we'd want to collect. So we're interested in the needles of pine trees, and we want to know if we can tell the difference between 2 species of pine trees by their needle length. Right. First, we want to go out and measure them and say, well, is there a big enough difference that we could reasonably use this measure? So Aleppo pines from Syria, we went out and we measured their needle length and on a bunch of different individuals. And then we calculated the average and standard deviation. So if we put that on our graph, we have this kind of pine tree, right? Aleppo pines, a species, and we have a different species over here, Torrey pines. So this is a categorical x-axis, right? And we want to know if we have these different kinds of trees, are their needles different lengths from one another? So again, we've measured the needle length of our Torrey pines. Torrey pines have really nice long needles. They are endemic, that is, they only occur in a small geographic area, to Torrey Pine State Park or State Reserve, which is just up the road from us, and also out on one of the Channel Islands. All right, so our very own Torrey pines showed up in our textbook, which is pretty exciting. Okay, so we've got the mean on here. The mean is about 10 centimeters for Aleppo pine. It's about 27 centimeters for Torrey pine. So we can see that the means are different from one another by about 15 centimeters. That's quite a bit. I can probably tell that difference by eye. And we also can see on this graph how variable the needle lengths are. That is, when we go and we measure different ones, are they all exactly the same size? Or are there a little bit of difference between them? Or is there a bigger difference between individuals? So we can see here with the standard deviations put on this graph, shown as these error bars that stick up and down, Aleppo pine not only is smaller, but it has the smaller differences between individuals. So the variation in the data set is smaller. Torrey pine has longer needles, and those needles are more variable in size from one another than the Aleppo pine needles are, but there's still a pretty big gap in between. Now, later on in the semester, we're going to be learning the statistical tests that we would use to say, are the needle lengths of these two pine trees significantly different from one another? And it turns out it takes into account both the difference between the means and how variable the data is around those means, right? We're going to make a graph that looks like this in our activity. We're going to call it a line graph. Now, we don't actually have these dots connected together with a line, so no line is shown, but a line graph is what Excel uses when we have a categorical x-axis and a quantitative y-axis. All right, so there's another neat kind of graph. We aren't going to make these in class, but I want you to be aware of them and know what they look like. So if we look at our line graph with our means shown as a dot and our standard deviation shown as these error bars, that's some information about the distribution, but we can actually make a graph that shows more information about the distribution, and that is called a box and whisker plot. And we have a box and whisker plot for the same data. And what we add to this representation of the data set is, instead of using mean and standard deviation, we're going to use median and then the quartiles. And we can extend the whiskers. So this middle part is called the box, and then these little extensions sticking up, kind of like the error bars, are called the whiskers. This is going to go to the maximum data point and the minimum data point. And the box is going to be made of the median, the first quartile, and the third quartile. So it's kind of like a little histogram turned on its side, right? And we can see that for Aleppo pine, the median is a little closer to the bottom of that box. That actually tells me that this data set is a little skewed, that it has a little bit of a right skew. All right, so this is just another way of representing the same kind of information. You're going to see these in papers sometimes. I'm not going to have you make them. Excel won't make them for us, so we aren't going to make them ourselves. All right. So again, how wide those distributions are is going to tell us something about how variable the data set is. Now, why does it matter how variable the data set is? This is an example of showing this bar here for group B. It has a really, really big error bar compared to these other bars. And this guy is saying, I'm sorry, man, but we just can't trust you. All right, so where we have a lot of variation, that tells us that the mean doesn't represent this data set very reliably, right? So there's so much variation between individuals that we just don't know a lot. We don't know as much about what the actual value of this variable is for this group as we do when the variation around the mean is much smaller. Right, so it doesn't mean that you did something wrong, you didn't inaccurately measure the data, but where we have that variation, where we have a lot of variation, it becomes hard for us to say something very clear and strong about what's going on in that group. Right, it might be the case that we actually have more than one kind of individual in this group, right? And we might want to investigate that and see what's causing all of this variation. What we're trying to do in statistics overall is understand why individuals differ from one another. We have put these individuals into groups A, B, C, and D because we think whatever this is, A, B, C, and D, is an important variable that will help us understand why individuals have a different value of whatever this y variable is, like whatever these group A individuals are. It's a smaller value than it is for these group D individuals. In that case, that variable, the difference between A and D, is explaining why some individuals are smaller and some are larger. All right, so remember, we're putting this explanatory variable on the x-axis. As you look at lots more examples of different data sets and you see what their means are and what their standard deviations are, you'll get a better idea of whether that standard deviation is large or small, and you'll feel more comfortable interpreting. All right, so as we go off to make graphs in Excel, Excel can do all kinds of things. That doesn't necessarily mean that we can. We don't want to have all kinds of goofy colors and patterns. We want to have it be clear. Mostly we want to have it in gray or black tones. Color printing is expensive. And so if we can represent the information well just using different colors of gray, black and white, then we definitely would prefer to. We want to have nice labeled axes, both X and Y. We want to have tick marks. We want to have nice, clear, large fonts that are easy to read. All right, so there is an assignment for you to do. There's going to be 3 different graphs to make, and there's a separate video showing you the steps to go through using Excel to create those graphs.